I think it's time to begin now. So welcome everybody to this molecular dynamic simulation webinar, and we are thrilled to have you, of course. So this is going to be another session of continuing from the previous. I'm not sure if all of you or any of you attended the previous sessions that we conducted, but I'm very hopeful that this is going to be a very interesting session for everybody, and everybody will have something to learn as well from here. So we, of course, have kept everything in the mind, that starting from the beginning and taking everything to the highest level as much as we can possible. So first of all, please let me introduce our company or our institution that we who actually helped us, and we are in a collaboration with the rest of our colleagues that helped in performing all of this webinar, which you are a part of right now. So we are actually called the Data Science Bioinformatics, or the Bioinfo experts. So we are actually an online bioinformatics company. My name is Arslan Hamid, and I'm a PhD in University of Bonn, Germany. And I'm bioinformatics by profession as well. And of course, I have other domains to work out. So of course, we are always very happy to help everybody in the field of the sciences as much as we can. Of course, from the data sciences, what we started is, we can definitely provide you the details or the help or any kind of instructions that you need regarding to the structural bioinformatics. So these are our expertise that I'm going to share with you right now, which we are dealing and which we are already complying with all of our collaborators and customers and other people as well, colleagues as well. So we are experts in the structural bioinformatics. We also can provide you with the services of the genomic sequences as well. We are also experts in transcriptomic services. And if we need it, we can also provide you with the AI or the machine learning based frameworks as well. We have the abilities to define and devise an algorithms from scratch as well, depending on your need. We can also provide consultancy services as well. For example, if you just need some help in your research, so we can definitely help you with any kind of consultancy services as well. And of course, training in the workshop. For example, you're part of this workshop right now. So of course, on demand, we can also do these kind of services as well. So by this note, please let me introduce you to the guest of honor today. He's the topmost collaborator for us, Professor Dr. Naeem Mahmood Ashraf. He's an assistant professor from University of Punjab, and he is expert in the protein engineering, bioinformatics, biochemistry, molecular biology, machine learning, algorithm development, and he has been doing many collaborations with us over a period of a lot of time. So formally, I would like to welcome Professor Naeem Mahmood Ashraf. Thank you very much for joining us today and giving us your precious time for this webinar. I am very hopeful that you will have something very interesting for everybody in our webinar today, and everybody will learn something and take something home. Thank you very much. Dr. Naeem, you have the con.
Thank you so much, Arslan, for having me here in this webinar and providing me an opportunity to share my experiences and my area of expertise with the other students and the researchers around the globe. And I wish you very good luck for your company, and I hope so that your company will add some value in the research work of your students, your customers, and your collaborators. So can you please allow me to share my screen?
I believe you can now.
Yes, thank you so much.
Yes.
So, dear participants, thank you so much for being here, and I hope so that this webinar will help you out to understand some basics of the molecular dynamics simulations. And not only we will focus on the theory in this webinar, we will also demonstrate practically that how you can perform the molecular dynamics simulations of a single protein or a simple protein in a water box. So the first thing which we are going to cover in this webinar is basic concepts behind the molecular dynamics simulations. And we hope so that after having this webinar, you will be comfortable to perform the molecular dynamics simulations by yourself if you have a computational power with you. We will actually focus, from the practical perspective, we will focus on the Gromacs. And this is one of the world best benchmark software to perform the molecular dynamics simulations. Obviously, there are the number of the different options that are available to perform the molecular dynamics simulation. Some are open source and some are purchasable. But I always love to work with an open source softwares because purchasable softwares, for the purchasable software, you have to buy the license, and after a certain period of time, you have to pay it again and again. Obviously, they are the user friendly, but I will always love to move with the open source softwares. And the Gromacs is one of the open source softwares, which is giving us a full leverage and the full potential to perform a different type of the molecular dynamic simulations on different type of the molecules. So right now we will just focus on the basic theory of the molecular dynamic simulations, and we will also focus on the molecular dynamic simulations of the protein. So let's begin and let's try to understand some very basics of the molecular dynamic simulations. Uh, keep one thing very clear in your mind, and that is what that all of the atoms which are going to make our whole universe, they are not static. In our textbook, in our BS textbooks, or in our graduation or undergraduate textbooks, we have seen the pictures of the atoms, and normally in these pictures you have seen the atoms on the molecules in a static form. But in reality, actually, the atoms are not in a static form. They are always having a jiggly wiggling moment. Even if you have a solid particles, in that solid particle, the atoms will have the certain type of the moments. Let's assume for a moment you have a brick. So this brick seems to be a solid, but actually this is and it seems to be static. But if you look microscopically into this brick, so then you will find it out that in this brick the atoms will have some jiggling wiggling movement, and the atoms and these jiggling movements are actually making our atom dynamic. The dynamic means that they are continuously changing their position in an x, y, and z coordinates. Similarly, let's say that you are going to heat the brick, and when you are going to heat the brick, so obviously the moments in the brick atoms will be enhanced, and because of these moments, there will be a flow of the heat from one part of the brick to another part of the brick. So let's say that when you are going to heat the brick, so the kinetic energy of the molecule, the atom, sorry, kinetic energy of the atom in the brick will be increased, and they will start to have a faster movements. Now, if you want to simulate it, that how these atoms are actually moving, and what will be their x, y, and z coordinates in the space after a certain period of the time when you are going to provide them the energy. So then we have only a one technique which is available right now in our hands, and that is going to be the molecular dynamic simulations. So in molecular dynamic simulations, we are going to simulate the behavior of the atoms after providing a certain force, certain energy, and certain type of the environment. Now, in molecular dynamic simulations, if this concept is clear to you, so let's talk about a bit more details of the molecular dynamic simulations. So in molecular dynamic simulations, if you want to perform, so then you need to have the two things to know about. The first thing which you need to know about, the first thing which you need to know about is the initial position of the atoms. And the second thing which you need to know about is the initial velocity of the atoms. So these are the two main and very important things which you need to know if you want to perform the molecular dynamic simulations. The first one is the position of the atom, the initial position of the atom, and then the initial velocity of the atom. And if you have an idea about the initial position of the atom and initial velocity of the atom, then after applying certain force on them, you can actually simulate it. Can you please make everyone mute, please? There's some certain
Yeah,
noise.
sorry, I just did. Yeah, I'm sorry. Please
Yeah.
continue.
Okay, so if you have an idea about the initial position and the initial velocity of the atoms, so then after applying a certain force, you can actually make a predictions that where will be these atoms after a certain period of the time. It will be just like that. Let's say that you have a one runner, and you know its initial positions, and let's say that you also have an idea about its velocity. So then you can make a prediction that where that runner will be after a certain period of the time. Let's assume for a moment you are driving a car. Let's take another example. Let's say that you are driving a car, and you are driving a car with the speed of 100 kilometer per hour. So we can actually make a prediction that after 1 hour where you will be, and how much distance you are going to cover if you are moving with the speed of the 1 kilometer, 100 kilometer per hour. So if you know the initial position and if you know the initial velocity of the atom, so it will be very easy to predict that where that atom will be after a certain period of the time. Along with these two things, another very important parameter which you know about it is the force, that how much force our atoms are going to exert on one another. So along with the initial velocity and the initial position of the atoms, the second very important thing is the force, because the velocity will be defined by the force. Obviously the atoms, if the atoms are going, if you are going to exert a more force on the atoms, so obviously they will have a more velocity. So another very important parameter, important thing to get an idea about the velocity is that we need to know about the force which the atoms are going to exert on one another. So that is another very important. But the question comes here that how you could have an idea that how much the force the atoms are exerting on one another, and how the force is going to define the velocity of the atom. So this is actually going to be a very good thing which we need to understand. To get an idea about the force, we need to know the interactions that the interactions which are present among the atoms. I hope so that in basic chemistry textbooks, you guys may have studied about the different type of the interactions that are normally present among the atoms. These interactions could be the covalent interactions. These interactions could be the van der Waals interactions, the hydrogen bonds, the electronic interactions, and etc. and etc. So if you have a one molecule and you know its initial and you know the initial position of the atoms of that molecule, and you know the what type of interactions which are present in them, so then based on those interactions you could get an idea about the force which the atoms in that molecule will exert on one another, and based on that force you can actually calculate that what will be their velocity. And based on all of these factors, you can actually get an idea, you can get some idea that where these atoms will be after a certain period of the time. Now, I hope so that you may have a good idea about these type of the interactions, the Coulombic interactions, the covalent interactions, the van der Waals interactions, and the hydrogen bonds. You may know it very well that the covalent bonds are formed due to the mutual sharing of the electrons. When the atoms come close to one another and then share their electrons present in outermost shells, which results the formation of the covalent bonds. And in organic in molecules which are going to make our life, the most common type of the bond is actually going to be the covalent bond. Let's take an example of the proteins. So in proteins which are made up of the 20 naturally occurring amino acids, there is going to be the peptide bond, and the peptide bond is basically a covalent bond which is formed between amine group and the carboxylic group of the two amino acids. Similarly, the Coulombic interactions are developed when the atoms come to a certain distance from one another, and then they're going to have charges on them, so it results in Coulombic interactions. Similarly, the van der Waals interactions, which are developed when the atoms are very close to one another. Another very important type of the interactions which are present among the atoms, these are the hydrogen bonds, and you guys may know it very well that the hydrogen bonds are basically developed between the electropositive hydrogen and highly electronegative element like highly electronegative atom like an oxygen, nitrogen, or the fluorine. I hope so that if you know the chemical structure of the water, so you know it very well that the water in the most dominating force in the water is basically the hydrogen bonds. And these hydrogen bonds are also developed when the electropositive hydrogen is actually at the certain distance from the highly electronegative element, and normally this distance is between 2.8 Armstrong to 3.2 Armstrong. So if you have an idea about all of these interactions, so then you can calculate the force. So a question comes here that how to get an idea about the force which the atoms are going to exert on one another. To get an idea about the force, I hope so that this graph will actually help you to understand this phenomena. So one thing, keep it into mind that when the interaction energy between the atom is zero, it means that there could be the two possibility when the interaction energy between the atom is going to be zero. The first possibility is that they are actually far away from one another. They are far away from one another. And the second possibility is that they are very close to one another. So keep it into mind that when interaction energy is going to be a zero, then no bond is going to be formed. And there will be the two possibilities as you can see here on your slides. When atoms are very close to one another, as you can see at the top of the slide, A1 and A2, when they are very close to one another, at that time the interaction energy will be zero and no bond will be formed. And the second type of the possibility is that when the atoms are very far away from one another, as you can see here on your right hand of the slide, A1 and A2, when they are far from one another, then obviously they can't make an interactions with one another, and at that time the interaction energy will also be the zero. When atoms are going to form in interactions, the atoms are going to form in interactions when they will be at an appropriate distance, and this appropriate distance is known as the R naught. And when the atoms are at the R naught and they are at the appropriate distance, at that time they are actually going to make then we are going to make an interactions. So the point I want to let you know, the basic point I want to let you know, if you have an idea about the interaction, that you know that how much they are actually far away from one another, from that point of view, you can actually calculate the interaction energy. And that interaction energy is basically will let you know about the force which the atoms are going to exert on one another. I hope so that now you will have a good idea about the force, that how you can calculate the force. Now, if all of these things will be clear to you, which I have just summarized, the first thing is the initial position of the atom. The second thing is the initial velocity of the atom. And to get an idea about the initial velocity of the atom, you need to get an idea about the force. And to get an idea about the force, you need to get an idea about the interactions which are present among the atoms of the molecules. So if all of these, can you please mute? Oh, thank you. So if you're clear about all of these things, so here at this point, we can actually apply one of the very famous equation of a Newton. And this is basically the second law of the motion equation. And this equation is letting us know that the force is equal to mass multiplied by acceleration. I hope so that you all will be aware about the second law of the motion, the Newton's second law of motion. This equation is letting you know that the force is equal to mass multiplied by acceleration. What does it mean? That if you have an idea about the acceleration of an object, and if you have an idea about the mass of an object, so what does it mean? You can calculate the force which that is actually going to play there. Now, let's say that I have an idea about the force and I have an idea about the mass of any object. So it means that I can calculate the acceleration. So do you remember that I told you that we need to get an idea about the velocity, and to get an idea about the velocity, we need to get an idea about the force. Now, if we have calculated the force which the atoms are going to exert on one another, and we have an idea about the masses of the atoms, so it means that we can calculate what? We can calculate the acceleration. And if we can calculate the acceleration, it means that we can predict it that where the atoms will be after a certain period of time. Please remember the same example. Please remember the example which I have quoted earlier about the quoted earlier, like if you are driving a car with a 100 kilometer per hour speed, and we know that where you are right now, and after 1 hour, we by looking at your speed, we can predict it that where you will be after a 1 hour. Obviously, you will cover a 100 kilometer per hour. You will cover the 100 kilometer distance after 1 hour. So please keep this point clear in your mind, that if we have an idea about the force that is playing between the atoms, and we know the mass, then we can calculate their acceleration, and that acceleration in which we are really very much interested in during the molecular dynamics simulations. So in molecular dynamics simulations, let's say that we have a one atom, and this atom is at the time 0. So we know its position and we know its velocity, and initial position and initial velocity, or you can say it as acceleration. So if I have an idea about its mass, if I have an idea about its mass and the forces which are basically interacting with this atom, so if I have an idea about the mass and the forces which are interacting on this atom, so then I can calculate its acceleration, and I can say that after a certain period of the time, let's say after a 1 picosecond, where it will be. Just like a car, you are driving a car with a 100 kilometer per hour speed. And now I know it that after 1 picosecond time, this atom will be at this position. Now, let's say that after 1 picosecond, now our atom is at this particular position, as you can see here on this slide. And let's assume for a moment, now I want to know that where this atom will be at after 2 picoseconds. Now I have an idea about the position of this atom after 1 picosecond, and I know that what above the forces are acting on that one. So I can make a prediction that where this atom will be after 2 picoseconds. And now let's say that I want to know that where this atom will be after a 3 picoseconds or the 4 picoseconds or the 1 nanoseconds. I know its position, I know its forces which are acting on it. So then I can make a prediction that where it will be after a certain period, or the 1 nanosecond or the 2 nanoseconds or the 100 nanoseconds. So this is actually known as the molecular dynamic cycle. So we know we try to calculate the position of the atoms and the forces on them, and based on them we calculate the acceleration, and based on the accelerations we are going to make a predictions that where they will be after a certain period of time in X, Y, and Z space, in the 3D space. I hope so that this point will be clear to you guys now. But here, there's one very important point which I want to talk about, and that is why we are going to calculate, why we are going to use a units of the nanoseconds or the picoseconds when we are going to conduct the molecular dynamics simulation studies. Let's say that you are in. I hope so that if you have read the papers, so in the papers, normally you will read that the authors have performed 100 nanoseconds molecular dynamic simulations of the protein. Question comes here that why they are going to perform a 100 nanoseconds simulation of the protein? Why they are not going to perform the one microseconds or the one whole for 1 second or for 1 minute simulations of the protein? Now, this is very important question, and we need to talk about it. Please remember, if we talk about the biological molecules, so for the biological molecules, the time period is not the seconds. The time is actually the nanoseconds or the femtoseconds in which the biological molecules are actually going to exert their action. If you will be confused at that time, if you will be confused in this concept, so let me elaborate it. Let's assume for a moment that you are in some threatening situation. And a threatening situation means that you are some in dangerous situation, and at that time, obviously, your fight and the flight response will be triggered. Now, if your fight and flight response is triggered, so then try to understand some basic biochemistry which is going to occur in your body system at that fight and flight response. What it will be, when you will see that dangerous situation, and your brain will send a signal to your adrenal gland, and your adrenal gland will release one hormone, and that hormone will be adrenaline. Now, adrenaline hormone will come into your bloodstream, and then it will bind with adipose tissues, it will bind, and the liver tissues, it will bind with your muscle tissues, it will bind with your heart tissues, it will bind with your lungs, and it will trigger the response. Now, let's say that your adrenaline is going to bind with the liver cells. So when it's going to bind with the liver cells, so then after binding the proprietary receptor at the liver cell, it will actually initiate a downward cascade, and in that cascade, what will happen? In that cascade, the glycogen will be broken down, and that glycogen it will be converted into the glucose, and then glucose will come out of the liver cells, and it will come into the bloodstream, and then it will be carried to the muscles, and inside the muscles it will be broken down to produce the ATPs. Now imagine for a moment that how much time is taken to perform that whole activity. It's just a 1 second in which that whole activity has been performed. It means that your enzymes, your proteins which are involved in that to carry out this whole mechanism are actually performing at not a 1 second or a 1 minute time period. They are actually performing their action in a picoseconds or a femtosecond. So that's why it's important to observe the behavior of the proteins, not at the seconds level, but at the nanoseconds level or the picoseconds level. And that's why in molecular dynamic simulation, if we are going to perform a molecular dynamic simulation of a protein, so it means that you should observe the behavior of the proteins, the folding of the protein, or the denaturation of the protein, or the binding of the protein with the ligand, not in a seconds time period, but you have to actually study it in a nanosecond time period or in a femtoseconds or the picoseconds time period. So that's why we are going to use here a units of the nanoseconds, where we are not going to use a units of the minutes or the seconds. I hope so that this point will be clear to you guys. If you guys have any question, then you can raise a hand and the moderator will allow you. So I will answer that question. Now, let's talk about some practical steps. So if we talk about the practical steps, so what will be the first step to perform a molecular dynamic simulations? The very first step, if you're dealing with the proteins, so then the very first step will be the preparation of the protein structures. Obviously, you need to have some protein, you need to have some structure to perform a molecular dynamic to perform a molecular dynamic simulation. So if in case of the protein, you need to have prepared your protein structure. The second step, once you will have the protein structure, once you will have the protein, so then there's in a second step, you need to prepare the protein topology file. What does it mean by the topology file? I will talk about this topology file very shortly in this webinar. The third step is that you need to solvate your protein and you need to ionize the whole system. Now, what does it mean by the solvation and ionization? I will talk about it in a bit later. After the solvation and ionization, the fourth step is going to be the energy minimization. You have to minimize the energy of the whole system. I will talk about it. And then you have to equilibrate your system. And finally, you have to carry out the production. And after the production, you are going to perform some analysis. So these are going to be the fundamental steps of the molecular dynamics simulation, starting from the protein structure preparations to the running the molecular dynamics simulation production phase. The production phase means that running of the molecular dynamics simulation. So these are some of the basic fundamental steps of the molecular dynamics simulations, and that these steps are going to be same. Whatever the software you are going to use, either it's a Gromacs or it's a NAMD or it's a LAMPS or any other type of the software which you want to use, these fundamental steps will be the same. There's not going to be the difference. There is not going to be different. Now let's talk about all of these steps one by one in bit detail. The first step is preparation of the three-dimensional structure of the protein. So whatever the molecule you have on which you will perform, on which you will want to perform the molecular dynamic simulations, you need to prepare its three-dimensional structure. Keep it into mind that in this whole universe, the objects are in a 3D shape. Either it's your table or it's your chair or it's your water molecule, they are always in a 3D shape. But obviously in our textbooks, we are going to see them in a 1D form, but in reality, they are in a 3D form. So if you want to have a realistic picture of their dynamic moments, so you need to have the protein structure or any other structure any other structure in a 3D form. So the very first step, if you talk about the proteins, you need to get the three-dimensional structure of the protein. The question comes in that how you can get the three-dimensional structure of the protein. So there are the two ways out through which you can get the three-dimensional structure of the protein. The first one is protein data bank. You can explore the protein data bank. I hope so that most of you guys may have a good idea about what is protein data bank. So you can explore the protein data bank and you can get the three-dimensional structure from there. And just very briefly, in protein data bank, in protein data bank, we are going to have the three-dimensional structures which are experimentally determined using these three experimental techniques. These three experimental techniques includes the X-ray crystallography, the NMR spectroscopy, and the cryo-electron microscopy. So these are the experimental techniques through which normally we are going to determine the three-dimensional structure of the protein. And all of these three-dimensional structures are available in protein data bank. You can go there and you can download your three-dimensional structure from the protein data bank in a PDB format. But if you're not going to find out your desirable structure in the protein data bank, so the second option is there for you. And the second option is that you can predict the three-dimensional structure of the protein. And to predict the three-dimensional structure of the protein, we normally use the three different type of algorithms. The first one is a homology-based algorithm, the most oldest one, and right now they are the outdated. And the second one is a threading algorithm. They are also outdated now, and I will talk about it very shortly. That why they are outdated, why they are outdated now. And the third one is the ab initio algorithms, which are basically the machine learning based algorithms. Currently, I hope so that you guys may have a good idea about the AlphaFold. So AlphaFold is a machine learning based algorithm. That's a revolutionary algorithm that is able to predict the three-dimensional structure of the protein with a lot of reliability. So currently in most of our analysis, if we are not going to have a three-dimensional structure in protein data bank, so we are going to predict the three-dimensional structure from the AlphaFold. But obviously, this is not easy to use an AlphaFold. So thanks to the AlphaFold, they have predicted the most of the three-dimensional structure of the proteins, and then they have deposited into a one database, and this database in AlphaFold database. So you can explore the AlphaFold database and you can download your three-dimensional structure by yourself from the AlphaFold database. Now, once you have the three-dimensional structure of the protein, so at that time, you can't take it straightforwardly to the molecular dynamic simulations. Particularly if you are going to download the three-dimensional structure of the protein into the protein data bank, so you can't take it directly to the molecular dynamics simulations. But why you can't take it directly to the molecular dynamics simulations? Because in most of the structures which are determined using X-ray crystallography, in those structures there may be some missing residues and the missing side chains as well. So if there will be the missing residues in the protein structure, so that we may create some problem when you will run when you will try them in a molecular dynamics simulation. So it will be very wise that once you download the three-dimensional structure from protein data bank, particularly those structures which are determined by X-ray crystallography, so please check it out that is there any missing residue, and if there will be missing residues, then please repair your three-dimensional structure before performing the molecular dynamics simulations. Similarly, if you're going to download the three-dimensional structure from of protein from protein data bank, so in those structures, most of time you are going to find out a lot of the non-proteinous structures. Like you may find out some water molecules present in the protein structure, some ligands, some metal ions. So these metal ions, non-proteinous structures and water molecules, they are not very much needed to observe the behavior of the protein. Sometimes they are co-crystallized when we are going to make a crystal structure of the protein. This concept may be difficult for you to understand, but anyhow, sometime during a three-dimensional structure prediction, we need to attach some non-proteinous structure with the protein so that we can get this structure. And obviously, they're not a natural part of the protein, so we need to remove them. So these two are a very key positions. These two are a very key position, key things which you need to remember before taking the protein structure to the molecular dynamics simulations. But if you have a predicted three-dimensional structures, particularly from the AlphaFold, then you can straightforwardly take them to the molecular dynamics simulation. But before taking these predicted three-dimensional structures to the molecular dynamics simulations, please be sure that your structure is accurately predicted. As you guys know it very well, AlphaFold is wonderful, but its accuracy is around 85%. The reported accuracy is around 85%. In case of homology modeling, the reported accuracy is just 25%. And in case of the threading algorithm, the reported accuracy is just 50%. So if you're using a threading algorithm, let's assume for a moment, so there are 50% chances are there that your structure could be wrongly predicted. So you need to just make it sure that your structure is accurately predicted. There isn't any flaws in your structure if your structure is predicted one. So before taking the protein structure to the molecular dynamics simulations, please be careful about these steps. Once you have prepared the protein structure, so the second step, as I told you earlier, is to prepare the topology file. So what does it mean by the topology file? So let me very brief. I will not go into in lot of details of the topology file, but let me very brief and let me try to be as simple as I can. The topology file basically contains all of the necessary information which is are required to run the molecular dynamics simulations. Now, please try to recall the previous part of this lecture. The previous part of this lecture, that I told you that we need to get an idea about the initial position of the atom. We need to get an idea about the force of the which is going to be exerted on the atom, and we need to get an idea about the mass of the atom. These are the three primary the primary informations which we need to have. Now, in topology file, you are going to have all of that information. You are going to let's say that in your protein there are 1,000 atoms. So you will have an idea about each atom mass, each atom, the force which are going to be exerted on each atom, and the initial position of each atom. Everything will be there. So that is actually going to be the topology file. So this file is going to be the heart of your molecular dynamics simulations, and this is going to be the topology file. Now, the topology file is normally prepared using a force fields. Now, this is again a very hardcore concept, force field. What does it mean by the force fields? So let me try to explain the force field in simple way. What does it mean? It's basically a domain of a biophysics, but for the biologist, let me explain it that what is a force field. So the force field is actually what? The force fields are basically the mathematical descriptors that normally used to prepare the topology file. What does it mean by the mathematical descriptors? The mathematical descriptors means that which are basically helping us to model that how the molecules, how the atoms in the molecules are going to interact with one another. at the atomic level. So these are the mathematical descriptors, but I'm not going into the lot of the details because it needs a huge amount of the time to talk about the force fields in detail. Primarily, these force fields are going to calculate the two things for you. Number one, the interaction energy, and the second thing is a force. Do you remember that I talked about the force in the start of this lecture, that you need to calculate the force? So the force fields are helping you to prepare the topology file. And in the topology file, there is going to be information about the force which the atoms are going to exert on one another. And you are going to have an idea about the initial position of the atoms, and also you are going to have an idea about the mass of the atoms. So you need to have the force field to prepare the topology file. So you have a protein structure, then you are going to use the force field, and by using the force field, you are going to prepare its topology file. Then topology file is going to have all of the information that is required to run the molecular dynamics simulations. I hope so that these 2 points will be clear to you. Arslan, is there any question up till now?
So far you're golden. You can go. I will let you know when there are questions. Yeah.
Oh, okay.
I think it's better if you just keep the questions till the end, so everything is smooth.
Sure,
it's my suggestion, of course. Yeah.
Sure. Okay, now let's talk about the force field a bit more. If we talk about in case of the Gromacs always comes up with some built-in force fields, and these are the 15 force fields which will be by default present in the Gromacs. Now, if you look at it here, that there is going to be there is going to be an OPLS force field, all atom force field. There is going to be the Embers force field. There is going to be the charm force field. So these are the three different classes of the force fields which are normally present by default in the Gromacs. Now, these force field will help you to prepare the topology file. But the question comes here that which force field will be appropriate for you? Now, this is something which is very important. So let me talk about it very briefly. The choice of the force field it totally depends upon the analysis which you want to do, that the protein structure which you have. Let's say that you have a protein structure that is bounded with the nucleic acid, and you want to simulate it that how that structure is going to interact with the nucleic acid. In this case, the third number force field will help you out. That is an Amber 96 protein nucleic Amber 94. That is actually the force field which will be most appropriate for you. Let's assume for a moment you have a one protein that is bounded with a ligand or a drug, and you want to simulate its behavior. You want to simulate the behavior of the drug with the protein. In this case, the charm force fields will be most appropriate for you, as you can see here at the 8th number. And let's say that you have just a simple protein, and you want to actually simulate its behavior. So in that case, you can use an OPLS force field. So I will highly recommend it to you guys that you should consult the literature before selecting the force field. That what type of the force field people already have used for the structure, which is just like your structure. So the force field, you will select the force field, and the force field will prepare the topology file for you. So once you have prepared the topology file and your protein structure is ready, so then you are going to solvate it and you are going to ionize it. So what does it mean by the solvation? Let's say that let's assume for a moment that I have this protein, this ball pen is my protein, and I want to simulate it. Now, as you guys know it very well, that the proteins are actually going to perform their action inside the water. They can't perform their action when they are dried. When they are outside the water, they can't perform any action. Water is mandatory. Water is necessity to perform the chemical actions. Chemical reactions. So what I'm going to do, I'm actually going to put this protein into this box, and this box is going to be what? This is going to be a water box. This is going to be a box, and inside the box, I will put my protein, and when I will put the protein, I will fill this box with the water. And this step is actually known as what? This step is known as a solvation. So we are going to put your protein inside the water. Why you are going to put the protein inside the water? Because you want to be as realistic as you can. And in natural environments, the proteins are going to perform their action inside the water. So once you have put the protein inside the water box, now the second step is that you need to ionize it. Why you need to ionize it? Because in a natural systems, in a natural cells, we have a neutral environment. So we need to ionize it. So why we are going to add the ions? We are going to add the ions so that we can make our system neutral. Because normally in the natural systems, we are going to have a neutral systems. After the ionizations, solvation and ionization, we are going to perform energy minimization. Now, this is very, very important, very, very important thing which you need to understand. What does it mean by the energy minimization? The one thing, keep it into mind that all of the proteins, all of the molecules which are present in this universe, they are always at the lowest energy state. If the molecules will be at the high energy state, they will be unstable. If the molecules are at the low energy state, they are stable. So every stable molecule which is around you, even the molecules which is going to make your bodies, the proteins which are going to perform the action in your body, and the proteins which are going to make the structures of your body, they are naturally at the lowest energy state. If they will be at the high energy state, they will be unstable. So I hope so that in basic chemistry textbooks, you guys have studied about it, that when we are going to provide the energy to the electron and we, on an outermost shell, the electron get the energy and they become excited and they jump from low energy orbit to the high energy orbit. But at the high energy orbit, the electrons are unstable, and that's why they release the energy which they have absorbed and they come down to their original orbit. Why they are coming back to their original orbit? Because at the high energy state, no one is stable, no molecule is stable. Now, if this theory is true, it means that the protein structure which we are going to take into the molecular dynamic simulations, we also need to make it stable before exerting any force on it. Before exerting any force on it, before understanding its molecular behavior, we need to assume it that our protein structure is stable. And if to make a protein structure our stable and make a whole system stable, what we are going to do? We are going to minimize its whole energy. So the stabilization of system is very important, because natural components, natural molecules are stable in nature. That's why we have to make our protein structure stable. And how we can make a protein structure stable? We can make a protein structure stable by minimizing its energy. And in energy minimization step, that's what we are going to do. We are going to perform, we are going to lower down the energy of the system. Now, if you want to load the energy of the system, so then normally we are going to use the two different type of algorithms. The one algorithm is known as the steepest descent method. Another one is known as adopted by Newton, Raphson, AB, and R. But most commonly used method is a steepest descent method. So normally in the publication you will read the
Sorry,
steepest
Dr.
descent
Naim, I
method.
think I think it's better if I ask the question right now, because otherwise it would be a moot point if I ask it later. There is a question in the chat: why to ionize the protein? So maybe you can answer
We
it
are
right
not
now.
going to
Yeah. Sorry.
we are not going to ionize the protein. We are going to ionize the system. We are going to ionize system. Right now, I'm just talking about with reference to the natural system. Inside the cell, there is going to be a neutral environment. So in the neutral environment, if you keep it into mind that we are not ionizing the protein, we are putting the ions in the system so that we can make the system, wherever protein is present, a neutral one. I hope so that this will be clear. If there be any question, then please let me know. I will answer it. Now, after energy minimization, the next step is going to be the equilibration. So in equilibration, what we are going to do, in equilibration, we are actually going to relax our whole system. We are going to relax our whole system. There's one more question like TIP30. I will talk about it when we will practically perform it. Now we need to perform the equilibration. After energy minimization, we need to perform the equilibrations. And normally we are going to perform the equilibration. We are going to perform the equilibration like by using NVT ensemble and NPT ensembles. NVT ensemble means in NVT ensembles, we are going to try to equilibrate the temperature, while in NPT ensembles, we are going to try to equilibrate the pressure of the system. Now, let's say that I'm assuming that my protein is behaving well at the 300 Kelvin. So I will try to make it sure before taking it to the production phase and the molecular dynamic simulation phase. What I will make sure that the whole system is at the 300 Kelvin, and the whole similarly in NPT, I will make it sure that the whole system is under 1 atmospheric pressure. So I will try to make sure that in equilibration steps, so that is actually going to be equilibrations in the two steps normally performed. One is NVT and otherwise is NPT.
Dr. Naim, there's only two small questions. I'm sure it's one-liner, so please, yeah.
Yes, please.
The first one is, how can we select a force field for a new system? Maybe you can answer this first, and then I can let you know the second one.
The selection of the force field that depends upon what type of the molecule you have. Let's say that you have a protein. So keep it into mind that the force fields are basically designed keeping the molecules in view. For organic molecules, we have a separate set of the force field. For the proteins, we have a separate set of the force fields. Let's say that I have a completely de novo protein. The de novo proteins means that that protein is not naturally present, but obviously that belongs to the protein family, and I know that, and there will be obviously there will be the 20 naturally occurring amino acids. So I have a set of the force fields which are specifically designed for the protein. Similarly, let's say that you have an organic molecules, and there are the certain force field which can actually you can use for all type of organic molecule. Even your organic molecule is going to be new one. Similarly, for the lipids, there are the separate set of the force field. For the nucleic acid, we have a force field. So you should know the category of your molecule, and if you know the category of the molecule, you can find out the appropriate force field for it.
Okay, perfect. The second question is, are you also adding sodium, potassium, and chloride ions to the whole system?
Yes, we are adding the appropriate ions into our system, and it depends that what type of ions we are going to add. Sometime we are going to add sodium, sometime we are going to add the potassium, sometime we are going to add the chloride. Now, during the practical steps, I will show the participants that how we can carry out this ionization step, and during practical step, I will let them know that we will actually ask our system, we will ask the Gromacs to add the two different times, the sodium and the chlorides. Now, sometime only the chlorides will be added, and sometime only the sodium will be added. Why? It's a Gromacs will decide that what type of the how many chloride ions and how many sodium ions, or only the chloride ions or only the sodium ions are necessarily to make a whole system neutral. Sometime to make a whole system neutral, only the chloride will be added, and sometime the sodium will be added, the positive ions will be added, and sometimes the negative ions will be added. So our objective is just to make a system neutral. It does not matter that what type of the ions there will be.
And I agree with Afiz as well. He's also suggesting the same thing that I was, that probably we can keep the rest of the question till the end of the presentation. But just moving to the last question you can answer from Sonia, and then of course we can probably keep the rest till the end of the presentation as well. How do we know that the force field constants are using in topology file chosen are correct? And after that, I'm so sorry, but we will definitely keep all the questions to the...
This is a really good question. This is a really good question, that how we could know that the force field contents are right. And for this one, we need to go through a good literature review. We need to understand that for which purpose that force field has been designed. We need to understand that the correct parameters are there for our molecule or not. So we need to understand, we have to go through the literature, we have to go through the force field parameters, we need to understand in a good way, and then we will decide that if this force field will be appropriate for us or this force field is not appropriate for us. Now that is going to be a very critical point, and that's a very good question which Sonia has asked. So you need to consult the literature, the appropriate literature of the force field to get an idea that these parameters are included or not, in which you are interested. Okay, so let's, any other question?
I think for now you can just move on, and we can keep the rest till the end of the presentation.
Okay, you can note it down, and you can ask all of those questions and participants at the end of this webinar.
Perfect. Thank you very much.
So okay, then after the equilibration, what we are going to do, we are going to perform a production stage, and in a production space, we will try to simulate that how our protein is going to behave in the system that we have designed in the previous steps. For a certain period of time, it could be 100 nanoseconds, 50 nanoseconds, 200 nanoseconds, 1 microsecond, or whatever. What is your objective? So these are the 6 basic steps which I have just told you. Now here, I'm assuming that because I'm a teacher and I know that sometimes the people are all the people are not at the same level. So let me introduce very briefly, before talking about the Gromacs, let me introduce very briefly that there are the 3 different types of the operating systems which are right now available in the market. The one is a Windows operating system, the most of us are actually using right now. And the second one are the iOS operating systems, the Macs, the Apple's laptops, and etc. and etc. And the third type of operating system is a Linux operating system. For the Gromacs, we need to have a Linux operating system. And why we need to have a Linux operating system? There are the certain advantages of the Linux operating system. And let me actually highlight few of them here for our participants. That the first thing, very important thing, is that the Linux operating system is an open source operating system. You don't need to pay any single penny to buy it. The second one is that it's a very secure and it's a very, very stable. And the third most important thing for the Linux operating system is that it's fast and efficient as compared to the Windows and Mac. And for this reason, the most of the heavy bioinformatic analysis are performed using the Linux environment. So I will highly recommend to our participants that if you are really interested to perform a bioinformatic analysis, I'm like molecular dynamic simulation, RNA-seq, NGS, and other type of the transcriptomic analysis. So you should have a good understanding of a Linux operating system and how to use it. And for the similar reason, we are going to use a Gromacs on the Linux operating system, because this is fast and this is efficient, and this will give you a better speed. This will complete your job in a relatively a shorter period of time as compared to the Windows or the Mac operating system. Okay, now let's move ahead and let's talk about the basic stuff about the Linux operating system. If you're going to use this Linux operating system, so then this will be a terminal window. So this terminal window, on this terminal window, we will type a certain commands, and these commands will be executed by the Linux system in certain programs to perform the action. I will demonstrate it to you. Now, as I told you earlier, that we are going to use the Gromacs. And the Gromacs is actually a software, or a benchmark software, I must say, to perform a molecular dynamic simulation. So if you look at the scientific literature, so in most of the research articles, people have used the Gromacs to perform the molecular dynamic simulations. Now, Gromacs is a one whole package, and inside this whole package, there is going to be the certain modules that we are going to use. And let me introduce, before going into the practical demonstration, let me introduce these modules to our participants. The first very important module that we are going to use inside the Gromacs is PDB to GMX. Now, what this module is going to do, this module is basically responsible to convert the protein PDB file to the topology file. So basically, this module, in simple words, this module PDB to GMX will help us to prepare the topology file by applying the force field in the Gromacs. The second module is a Solvate module. And the Solvate module will do what? This will carry out the solvation, as I told you earlier. The third module is the Gromap. The Gromap is a very important one, once again, and this will actually prepare the atomic structure input file, and this file will have an extension of TPR. I will show you this file when it will be generated inside the system. Another very important module is a GenIon, and this GenIon module is basically carry out the ionization. It will make the system neutral by adding the ions, either cations or the anions. And another very important module is an MD run, and this MD run will be used to carry out the molecular dynamic simulations, and this MD run will be also used to carry out the energy minimization and as well as the equilibrations. So I hope so that these modules will be clear to you. So let me repeat them once again: the PDB to GMX to prepare the topology file, Solvate to carry out the system solvation, Gromap to create the atomic structure input files, so the TPR files. The next one is a GenIon for carry out the ionization and the neutralization. And the MD run module to run the molecular dynamic simulations. I hope so that this will be clear. This will be the last point, and after it, we will jump to the practical demonstration. Now, the first step, I'm going to talk about the input and the output, which we will have in each step. And I hope so that if you have a good idea about this workflow, it will be easy for you to understand the practical demonstration. So in the first step, we will give our PDB file, or the protein three-dimensional file, and we will have an output. Sorry, forget about the first step, come to the second step, sorry. The first step is not mandatory. Now, in step two, we will give our PDB file, and the output will be GRO file, and the topology topl.top file. They're the two files which we'll have as an output. So these are the only file where I'm emphasizing. There will be multiple other files will be also created, but these two files will be most important one in an output of a step two. The first one is we will give the PDB file, and then we will have the GRO file and the tuple.top file. The tuple.top file will be basically the topology file. And GRO file is basically the three-dimensional coordinates of the protein structure compatible for the Gromacs. In a third step, step number 3, what we will do, we are going to solve it. After preparation of the topology file, now we are going to solve it our system. Now to solve it our system, we will use the GRO file that is created in a step number 2, and then we will take it as an input. And then this solvation, in the solvation step, on a step number 3, we will get another file, GRO file, and that will be the solvate file where the solvation has been done. And the tuple.top file, which is a topology file, that will be updated. The tuple.top file will not be created again in this step, but the previous tuple.top file will be updated. In step number 4, which is ionization step, we will use the GRO file that is created during a solvation step, and we will use it as an input. And then in this step, we will have another output of GRO file where we have a proper ionization of our system. And the tuple.top file will be once again updated. The topology file will be again once updated. In step number 5, we will use the test_ions.gro, the ionized GRO file as an input, and we will get another output of em.gro. And em.gro will have the energy minimized system. The energy minimized, that will be energy minimized step. The step number 5 is energy minimization. And once again, the tuple.top will be updated. I'm talking about the reference of Gromacs. In step number 6, which will be obviously the equilibration step. In equilibration step, we will take the previous energy minimized file, em.gro, as an input, and then we will have a 2 modes output, npt.gro and npt.gro. And once again, the tuple.top file will be updated. And finally, we will take the equilibrated GRO file as an input, and we will take it to the MD simulations. And at the end, we will have one file which will have the trajectory, which will have our whole data. What does it mean by trajectory? I will talk about it. Which will have our whole data. That will be an XTC file. So this XTC file will be analyzed at the end to get the data, to get the results. Now, what type of the results we will have? We could have a 4 different type of, normally the 4 different type of the results normally we are in which we are normally interested. The first type of the results in which, first type of the analysis in which we are interested, that is a root mean square deviation, normally known as RMSD. The second one is known as RMSF, root mean square fluctuation. Third one is the RG value, or the radius of gyration. The fourth one is a solvent accessible surface area, normally known as SASA. And the fifth one is total number of the hydrogen bonds. Right now, the graph which you are going to see it here, that this graph, right now this graph which you are going to see it here, that this is an RMSD graph, the root mean square deviation graph. On an x-axis, you can see the time in a nanoseconds. On a y-axis, there is a root mean square deviation values in an Armstrong. Now, what this root mean square deviation is letting you know, the root mean square is basically letting you know that how much your structure has been changed over the period of time. And if you look at this graph in bit detail, then you will see that at the 0 nanosecond, you can see the red color line near at the 0. There is but as the time is actually progressing, the protein structure is getting changed over the period of time, and you can see the increased RMSD values. How these RMSD values will be calculated? These RMSD values will be calculated. Let me show you here. Let's say that this is going to be your initial protein structure. And this is actually going to be your protein structure after 6 nanoseconds. So what we are going to do, we are actually going to compare this structure. We are going to compare this structure with this initial structure to get an idea about the change that is happened in the protein structure after a certain period of time. And this is actually known as the root mean square deviation. So we are basically observing the deviation of the structure as compared to the initial one over the period of time, that is known as RMSD values. In a second graph, you can see it here, that you can see the 2 different RMSD curves. So let's say that this is the one RMSD curve, and this is going to be the second RMSD curve. And let's say that this first RMSD curve is protein A, and the second one is the protein B. So let's say that the second one is a protein B, and we have actually run both of them in the molecular dynamic simulation over the period of 30 nanoseconds. So you can see it here that the one protein is giving us a higher RMSD values, another one is giving a lower RMSD values. What does it mean? It means that the protein B is changing its structure more over the period of time as compared to the protein A. So this is an RMSD, and that's what the insight we could have from the RMSD. Another type of the graph in which people are normally very much interested, that is the root mean square fluctuation. And this graph is basically helping us for what to get an idea about the fluctuations in our protein structure. On an x-axis, you can see the amino acid residues, and on a y-axis, you can see the root mean square fluctuation values here. So what this graph is letting me know? This graph is letting me know that approximately first 200 amino acids are showing a higher fluctuations during the molecular dynamic simulation. The higher fluctuation means that they are showing a higher kinetic energy. They are not a well conserved, well bounded structures in a protein. They may be loops which are actually moving a lot during the molecular dynamic simulations. While this area of the protein, starting from around 300 amino acids to the 800 amino acids, you can see here that the peak size is not very high. So what they are letting you know, it means that in this area, there is actually not a very much high kinetic energy. So this could be the core of the protein. And once again, if you look at it here at this area, so you can see that there is a lot of the kinetic energy here. So you can see that one from which amino acid to which amino acid this area is having a high kinetic energy. So this is another very important insight that we could have from the molecular dynamic simulation data. Another graph, which you can see it here right now, this is a radius of gyration. On an x-axis, we have a time. On a y-axis, we have a radius. And what does it mean by radius? I hope so that most of you guys would have a good idea about it. So let's say that this is our protein, globular protein, and this is going to be the radius. And over the period of time, obviously protein needs to change its conformation, and if protein is going to change its conformation, so its radius will be increased. Its radius will be increased. So that is actually going to be the radius of gyration. So you can see it here that in the starting, we have a protein structure radius of 4.2 nanometers, but over the period of time, the protein has changed its conformation, and now radius is 4.5 at the end in this graph. That's what the interpretation we could have. Another one is the total number of the hydrogen bonds. You know it very well. The hydrogen bonding is one of the most important that is a dominant force to stabilize the protein structures. So here on an y-axis, you can see the total number of the hydrogen bonds, and on an x-axis, you can see the time. So here we can actually monitor it that how many hydrogen bonds have been broken down or formed over the period of the time. So if this slope is getting up, it means that more hydrogen bonds are formed over a period of time. If this slope is getting down, it means that the number of the hydrogen bonds has been reduced over the period of the time. And the last type of the graph, the fifth one, is a SASA, the solvent accessible surface area. So as you know it very well, your protein is present in a solution. Your protein structure is present in a water box. So its area in SASA, in solvent accessible surface area, we are going to calculate the area of the protein which is accessible to the water molecules, where the water molecules or the any solvent that is interacting with the proteins. That's what the SASA is, the solvent accessible surface area. So if the solvent accessible surface area is getting down, this curve is getting down, it means that the protein solvent accessible area is going down. And if the solvent accessible area is getting up, it means what? It means that your protein is most likely going to be denatured because it's interacting more with the water. The denatured proteins interact more with the water as compared to the properly folded proteins. So I hope so that these three graphs will be clear to you. So I will take just a break of 2 minutes, and this in this break, I hope so that the moderator has said something to you guys. So let me prepare myself for the practical demonstration. And after the practical demonstration, we will have a proper question answer sessions. We will answer all of your questions that you will have. So over to you, Arslan and Hushbak. So thank you so much for this theory portion. Now let me prepare myself for the practical demonstration.
Thank you very much, Dr. Naeem. Of course, I see many interesting questions in the chat so far. And of course, definitely we will be answering. And of course, we will try to answer all as much as we can. And Hushbak, please, over to you.
Thank you, Arsalan. I hope, you guys have learned a lot during this session, and I know you have some questions, but I'm gonna take this opportunity to introduce Bioinfo experts to you. I hope my screen is visible to you all. Can you guys see it?
Yes, we can.
Yes.
So, amazing. So we started Bioinfo Expert the idea came in 2020. So we started a YouTube channel, and so we thought we can share the information with everyone on bioinformatics, MD simulations, molecular docking, and everything. Later, based on the positive responses, we extended our wings to Udemy, and then on Fiverr and LinkedIn. This is just a little introductory page of our YouTube channel, where currently we have more than 4,600 subscribers, and we have uploaded 61 videos that provides information on how to perform molecular docking, build phylogenetic tree, how to do molecular dynamic simulation, or how to predict protein structures, and many others. By the grace of Allah, we have more than 200,000 views on YouTube channel, and we have more than 200,000 views. And based on that, we decided to extend our wings to Udemy. Now, Arsalan, how much time do we have? Should I continue, or should I proceed to Dr. Naeem?
You can take a time. I just need one more minute to complete my preparations.
Okay, so after seeing the success on YouTube, we rethought and we decided to make some objectives for Bioinfo experts, and we decided knowledge is power, and we should take steps so we can expand and we can teach society among us and explain them how important is bioinformatics for the society. So we started 2 courses on Bioinfo experts, on Udemy. And these are the QR codes for Udemy courses. If anyone is interested to learn, they can scan these QR codes. And I will also share the URL for these courses in the chat. And if you guys contact with us through LinkedIn or any other means, we also provide private consultancy, and we can also give you some discounts on these courses on Udemy. And you can see people, they have really benefited from these courses, and you can see we have gathered very positive responses over the time. I'm sharing some of the recommendations with you. And as for today, from 2020 to 2023, we have students from all over the world in Udemy, and most of the participants, they are from Asia, Pakistan region. They are from Europe, and we have also participants from the United States and far-off regions, and they are all extensively learning from this course. So this is it from now, and I will again remind you guys to please register, mark your attendance by scanning this QR code, so we can, after 2 and 3 days of this session, we can give you certificates. So this is from my side, and Arslan, over to you.
Yes, of course. Afiz, I see your this. I think you have a question, probably. I don't know.
Yes, I have a question. I thought the professor has completed the presentation.
No. Actually, now I think we're going to be moving into the this practical session of the webinar. First, of course, the first portion was for the theory. Now he will explain exactly how to perform this molecular dynamic simulation, as he explained earlier.
Okay.
Perfect. Awesome. Over to you, sir, Dr. Naeem.
Oh, okay. Thank you so much. I hope so that you can hear me. So I hope so that let's start this practical portion. So right now we are going to bring here a very small protein. As you guys know it very well, obviously the proteins are going to be the large macromolecules, but right now, just for the sake of demonstrations, we are bringing here a very small protein, and this protein is just a 30 amino acid long. As you can see it here, that this is just a 30 amino acid long protein, and currently I have opened this protein into the PyMol. So just to let you guys know that what type protein is. You can see it here that in this protein there are the 2 beta sheets, and there is going to be one small helix, very small helix, and there is going to be a 1 loop. So we are actually going to simulate this protein in our system. I was reading the questions during that preparation, so I will answer all of those questions. Please don't worry. Let me complete this simulation. So as I told you earlier that we are going to use a Gromacs. So for the Gromacs, we need to have a Linux system. So normally what I'm going to do, normally I'm going to prepare all of the necessary files in my own system, and then I will shift them to the supercomputing facility where the normally molecular dynamics simulation is performed. Because molecular dynamics simulation is a very computationally expensive job. There will be tons of calculations that have to be done. Keep it into mind that in a one protein there could be thousands and the thousands of atoms, and we have to keep track the information of each and every atom. So if you are calculating the position of each atom after 1 femtoseconds, so then you can imagine it that for thousands and the thousands of atoms, how many calculations will be done. And for over 100 nanoseconds, how many calculations will be done. It's going to produce GBs of data. And sometime once during my academic study, I was doing a one molecular dynamics simulation of a protein, and that protein produce approximately a 30 GB of the data. That's very normal. That's a huge amount of the calculations are going to be carried out. So that's why you need to have a good computing power. So right now I'm going to prepare.
Yeah, we have produced 70 to 80 gigabytes per complex as well. So it can be huge, of course.
Yes, and right now I'm going to prepare our protein files just for the sake of demonstration. A very small protein in my Windows system, and then I will take it to the supercomputing facility where the normal production phase will be run. So in my Windows, as you know that this is a Windows operating system, which is right now you can guys can see it here that. So I have installed Ubuntu in my system. And so let me show you here. So let me go into my terminal and type in Ubuntu. So you can see it here that this orange color sign, this is a Ubuntu which I have installed in my system, the Windows system. So this is known as WSL. So what you can do that you can use this facility in the Windows, the Ubuntu facility in the Windows, and you can actually use the Linux-based software by using this Ubuntu WSL. By the way, in the new Windows, this facility has been built in. But if you are using a Windows 10, so then you have to need to install, and then you need to install the WSL. Please keep it into mind that those guys who have a Windows 8, the Microsoft has disabled the support of the Windows 8. So if you have Windows 8, you can't install this WSL. So please update your systems to use the WSL up to the at least the Windows 10. If Windows 11 is, then it will be perfectly fine. So how you can install this WSL, the Ubuntu, in your system? It's a very easy job. So what you need to do, you need to go to your Windows Store, the Microsoft Store, and in this Microsoft Store, please search the Ubuntu. And when the Ubuntu will come in front of you, then simply click on the Get button, and the Ubuntu will be automatically installed in front of you. So right now, you can see it here that I'm in our Windows Store. So in this Windows Store, I will type here the Ubuntu. I will type here the Ubuntu, or you can use any type of WSL. I like the Ubuntu, so that's why I'm going to use it. So when this will come, so you will have a lot of the suggestions related to the Ubuntu. Yeah, you can see it here. So you can select the most appropriate for you, obviously the most latest one, and then simply click on this install button. And when you will do that, this WSL will be installed in your system. When the WSL will be installed in your system, so what you will do, we will go to the start menu, and then we will click on this one, and this background window will appear in front of us. And right now we are in our root directory. Obviously, you may take some time to familiarize this command line system, because normally we are used to do some clicks on the folders, but here we need to use some commands to navigate to the folders. So the very first thing is that you need to install the Gromacs in it. Now, how you can install the Gromacs? I hope so that the moderator of this meeting will share one document with you, and through that document you will have a good idea that how you can install the Gromacs in this system. But obviously I'm not going to do it right now because it will take a huge amount of the time. So I'm not going to demonstrate that how you can install the Gromacs. So let's move ahead, and we have installed the Gromacs in our system. Let's move straightforwardly move ahead. So the first thing is that we have to move to the folder. And we have one folder here, which you can see it here that, and I actually named this folder as a web Webnair test. And this folder is located in my D drive. So I have to go to the D drive and then from the D drive, I have to go to my web test. So what I will do, I will come to this window, the WSL, and I will type cd/mnt. MNT is what? It's hard to define it right now, but keep it in mind, you have to use this MNT. So as you can see it here, that the blue color, from my root directory, right now I'm in MNT in a blue color. And then from if I will type the ls is actually command to check the content of the directory. So I will type an ls. Then you can see here that right now in my system, there are the 3 drives, C drive, D drive, and the E drive. So where I need to go to the D drive. To move to the D drive, I will use a CD. The CD is a command of change directory. So I will type CD, change directory, where I want to go to the web test. So I will type the name of my directory, which is a web test. So CD space web test. I'm actually asking the Ubuntu, please move to the web test. So right now, I have to first move to the D drive. So I will type CD and then I will type D. And I will hit the enter button, and you can see it here that along with the MNT, you can see the D as well. We are getting short of time, so that's why I will be a bit quick. Once I will be in the D drive, so I have to move to my test folder, and this test folder is a web test, as I told you earlier. So as you can see it here that right now we are in a web test. Let's check the content of this directory by typing the ls. So I have typed the ls. And you can see it here that there are the 2 files right now. The one is 83.pdb file, in which I'm obviously interested, for which I want to perform a simulations. And the second one is going to be the force field file. Now, the very first step, as I told you earlier, and most of guys will have a good idea about it, that we are going to prepare the topology file. So how we are going to prepare the topology file? To prepare the topology file, we are going to use one module of a Gromacs, and that module is PDB to GMX. So what we will do, we will type GMX. The GMX is basically abbreviation of a Gromacs. So you will type GMX. The GMX, basically you are asking Ubuntu that Ubuntu please use GMX software. It means Gromacs softwares. So after typing the GMX, we will give a space and we will type the name of module in which we are interested. And we are interested in a module PDB to GMX. Do you guys remember that I told you that this module will actually take the input of your PDB file and prepare the topology file for you? So after typing the PDB to GMX, I will give an input of my file in which I want to use. Then to give an input, I will type -f, and after typing -f, I will type 83.pdb. 83.pdb, that is actually going to be the name of file that I have. After typing the input file, I will type -o, and the -o is going to define my output. And my output, I want to have an output, let's say that I want to have an output as test.gro. The test.gro, I want to have an output. So after typing this, what I will do, after typing this, I will simply hit the enter button. And as I will hit the enter button, you can see it here that the list of all of the force fields come in front of me. You can choose any force field depending upon your need. If you guys remember, then I have placed one more folder here. I told you that in a theory part, that there are the 15, Gromacs comes with the 15 built-in force field. But if you are interested to use any other force field which is not present in a Gromacs, so what you need to do, you need to put the folder of that force field in your working directory. In your folder, you need to place your force field folder. So when you will place your force field folder, then what will happen? Look at the first one. This one is actually the force field. This one is actually the force field, which folder we have placed in our working directory. So if you want to use any of the force field in which you are really interested and that force field is not present in the Gromacs by default, so then you can put this folder of that force field in your working directory, and that's it. Let's assume for a moment right now we are interested in our force field, which is a force field 1. So what I will do, I will just click here. I will type a 1 here. 1 means that I'm asking it that please select the first force field. And then I will hit the enter button. And as I will hit the enter button, now I'm going to have an options. Now these options are what? These options are TIP 3P, TIP 4P, TIP 5P, SPC, SPCE, and none. what these options are. These are basically the water models. And by default, normally TIP3P water models are used. By default, there's a recommended one. You can see it here that the Gromacs is by default recommending it that you should use TIP3P. If you are using TIP4P, then more computational power will be used. If you are using TIP5P, then the even more computational power will be used. So normally we are going to run the molecular dynamic simulation on TIP3P. The TIP3P are basically the 3-point contact of water. It will be hard for me to explain all of that things right now in a 1-2 hour session. So it needs multiple sessions, and I hope so that the Bioinfo expert will arrange the multiple sessions to make it more advanced, and you will join us in the next session as well. So TIP3P is basically the water model system. So TIPs 3P, 4P, they are water model system. Similarly, the SPC, the single point charge, the simple point charge. But mostly people are going to use the TIP3P, the 3-point contact charge. In 4P, 4-point contact charge is used, and 5P, the 5-point contact is used. So right now we are going to use TIP3P. I will hit the one button. I will press the 1 because in this menu, I need to actually select the first one. So after typing the 1, I will hit the enter button and look at it here that we faced an error. And what is this error? This error is suggesting us that serine 1 was not found in RTP entry. It means that there is something wrong with our protein structure file, so don't worry about it. We are going to use one more argument, -IGNH, and IGNH stands means what? We are asking the Gromacs that please ignore it. So once again, I'm going to repeat all of those steps. And here we go. The Gromacs prepared the topology file. And the one very important thing, or a bit funnier thing for the Gromacs is that when the Gromacs is successfully executing your command without any error, so it will give you one quotation. And you can see it here that the last line, the Gromacs reminds you, everybody wants to be naked and famous. And that's what the one quotation that the Gromacs is letting us know. So that's this quotation means that your command has been successfully executed. Now let's check the content of our directory once again by typing the ls. The ls, as I told you earlier, is actually the command. So this is actually going to be the command. So check the content of the directory. So you can see it here that some more files has been created. Let me go back to the directory and let me show you the files. So you can see it here that before execution of this command, we have just 2 files here. The one was our force field folder, another one is 83.pdb. But here you can see some more files has been created. The 2 files are pretty important here. The first one is a test.gro, another one is topl.top, as you can see it here. The topl.top and the test.gro file, these are the 2 most important ones. So our topology file has been created. Let me show you the topology file once again. I'm going to open this topology file in a notepad. I will highly recommend it to you that please don't change anything here. If you're going to make any tiny change in this file, this will actually bring an error in your subsequent steps. So you can see it here that in this topology file, you are going to have information about the charge, you are going to have information about the mass, you are going to have information about the atom, the residue, where this atom is present. This atom is present in serine. This is part of this group. The atom is nitrogen. Cg charge is -0.3. Mass is this, and rest of the information, as you can see it here, that look at this one. This is the content of the topology file. So this is having all type of information which is required to run the molecular dynamics simulation, and this is prepared with the help of the force field that we have selected. Now, after doing it, the second step is going to be what? After doing it, the second step is to solve it. Now, solve it step will be done in a 2 sub-step. There are the 2 sub-steps to perform the solvation. The first step, we are going to create a box. And to create a box, once again, I will type here GMX. I will ask Ubuntu that please use GMX. Then I will type one more module name, and this module name is edit.conf. After typing edit.conf, I will give the input file name, and the input file name will be what? I hope so that you guys will remember. That will be an output of our previous file, previous command. The output of a previous command is test.gro, so we will type test.gro. Then we will type the output file name that we want to have after execution of this command. and let's say it test underscore new box, because right now we are not solvating it, we are just creating a box just like this one. I'm not filling this box right now with the water, I'm just putting my protein right in this box. So this will be the name of my output file. Then I will give a one argument of -c. Then I will give a one argument of -d. After -d, I'm going to type 1.0. What does it mean by -d? The -d is going to define the dimensions of the box. Obviously, every box is going to have a certain dimensions. The 1, 1, 1, 1, 2, 2, 3, 3, whatever. So through this one, I'm actually going to give the dimensions of the box. And then I'm going to type BT. The BT stands for the box type. And here I'm going to write the cubic. I want to have the cubic box. I hope so that you guys will be familiar with the cubic. So the Gromacs will provide you a different options, like you can create a rectangular box, you can create an octahedron box, you can create a cubic box, and you can create a multiple other type of the boxes. You can check the Gromacs documentation for more information that what type of the boxes you can create here. But the most commonly used box is actually going to be the cubic box. So after typing this command, we will hit the enter button, and you can see it here there. Look at the output that our box is created. Now our protein is in the box, but still the water is not there. Now we need to add the water. And to add the water, I told you earlier, sorry, to add the water in this box, having the protein right now, we are going to use a solvate module. To use a solvate module, once again, we will type GMX. We are requesting the Ubuntu that please use a GMX, and then we are going to use a solvate. Please be mindful that spelling mistakes are not allowed here. Then after this one, we will type -cp, and -cp, we are going to give an input of our previous file, and our previous file is the test underscore new box.gro. After typing this one, after giving an input, then what we are going to do, we are going to give an argument of cs, and then we are going to give spc216.gro. What is this spc216.gro? This is a bit hard for me right now to explain it because it needs a lot of details. So that's why, just to save the time, I'm going to just skip it. And after this one, we are going to give a -o argument, and -o argument is going to give the name of our output file. We are going to type a name of our output file, test solvate.gro, and then I will type -p and the tuple.top. Tuple.top. Do you guys remember I told you that in every subsequent step, the topology file will be updated. So here we are going to ask our Linux Gromacs that please update. -p tuple.top is basically letting the Gromacs know that now Gromacs has to update the topology file. So after typing this command, we are going to hit the enter button, and as you can see that the solvation has been done. And here in this output, you can see that how much water molecules have been added, what is density. So total 13,408 water molecules has been added in the box, and the volume of this box is this one, and the density of the box is this one. You can see it here that. Now after the solvation, the next step is going to be ionization. So how we are going to perform the ionization? So let me explain it to you. And here I hope so that the guy who has asked a question that what type of ions we are going to add, I'm going to elaborate here in this step. So once again, we are going to type GMX, and here we are going to use a Gromap module. Gromap module. I told you earlier that the Gromap module is going to prepare the TPR file for you. So for the ionization, energy minimization, equilibration, and production, you need to have the TPR file. So first you will prepare the TPR file, and then you will carry out the ionization. Then you will prepare the TPR file, then you will carry out the energy minimization. Then again you will prepare the TPR file, and then you will carry out the NBT and NPT. And then you will prepare the TPR file, and then you will carry out the MD simulation. So TPR file is very important one. So Gromap module is going to prepare the TPR file. So once again, here we are going to provide the Gromap, and then we are going to give an input of file. And this input of file is ins.mdp. Now, what is this ins.mdp, and for where you can get it? So let me search it here. Arslan, can you please send me the file, please? I hope so that I will have here. Yes, I have here. Okay. No, I don't have here. Where is ines.mdp? I'm sorry. Yes,
Should
this
be
one.
outside, yeah.
Yes, this one. So I'm going to copy this ines.mdp file and I'm going to bring it here to the web test. So what is the MDP file? The MDP stands for molecular dynamics parameter file. So let me show you this file. I'm going to open it into a notepad, and here you can see the contents of that mdp.mdp file. If you guys will be interested, then we can, I hope so that the moderator of this session will send these files to you as well. So you can see the content of these files. You can prepare it by yourself if you are expert one. So you can also read the Gromacs tutorials or Gromacs module, the Gromacs documentation, that how you can prepare this MDP file. So this is one of the most important files that you need to have for the molecular dynamics simulations. So we are going to give an input of ines.mdp, and then we will type -c, and then we will give the input of the previously created file, which is test-solvate.gro. After typing is, once again, we will type -p, sorry, we will type -p, and we will type topl.top. As I told you earlier, that once again we are going to update the topology file, and then we will type -o, and we will type the name of the file which we want to have in response of this command, and this file is ines.tpr. As I told you earlier, the group member module is going to produce a tpr file for you. So this is actually going to be a command, and after having this command, after typing this command, we will hit the enter button, and tpr file creation will begin. And you can see it here that the tpr file has been created. As you can see it here that the Gromacs reminds you all you need is greed. I don't know who is that one. So, but this is going to be a quotation at the end of successful execution of the command. Now we are ready to add the ines in our system. To add the ines in our system, what we are going to do, let me show you here. We are going to use a genine module, genine. We are going to use a genine module, and then we are going to provide ines.tpr as an input. And then what we are going to do, we are going to define our output -o test_solvate _ines.gro. That's going to be our output. Then once again, we are going to update our topl.top file, and after updating a topl.top file, now we are going to pname. We are going to type pname. What does it mean by the pname? The pname stands for the name of the positive ions. What type of positive ions you want to add? Maybe you can add a potassium, you can add a calcium, you can add a sodium. So right now, let's say that I'm interested in a sodium, so I will type Na. The Na is a conventional name of the sodium, but you will type it in a capital letters, not a small letters. So Na, these are the positive ions which I want to add. You can add a potassium, you can add a calcium, whatever you want to use. Then I will type N name. The N name is actually what? -N name is going to define the negative ion names, the anion names. And here I am interested to provide the chloride ions. So that's why I'm typing the Cl in a capital letter. And after it, I'm going to type one more argument here, and that is neutral. Spelling mistakes are not allowed. Neutral, neutral. What I'm going to say by this neutral? I'm going to ask, or I'm going to request the Gromacs that Gromacs please add the sodium and the chloride ions in the system, but please make the system neutral. Add the amount of the ions which is required to neutralize a system. After typing this, I will hit the enter button, and then I will have this menu. In this menu, it is asking me that where I should add ions. I should add ions in system, I should add ions in proteins, I should add ions backbone of the protein, where I should need to add ions. Now keep it in mind that we want to neutralize the system, we want to neutralize the environment where protein is present. We don't want to ionize the protein. So if you are going to select the one here in this menu, if you are going to select the one here, so it means that the protein will be ionized. But our objective is not to ionize the protein. Our objective is to ionize the solution where the protein is. And you can see it here that at the serial number 13, if you see here, then there is going to be SOL. The SOL stands for what? The SOL stands for solution. So I want to neutralize a solution. So that's why I'm going to I'm going to type the 13, and I'm going to hit the enter button. As you can see it here, Dad. Now look at the output. That was a question of from one of the participants. And look at the output. Now in this output, you are going to have an idea. Only 2 chlorides are added. But if you remember the command, so in the command we have asked the Gromacs to add the sodium and the chloride, but the Gromacs only added the chlorides, only added the chloride, not the sodium. Why? Because the Gromacs calculated by itself there to neutralize the system. I just need to add the chlorides, nothing else. After doing this, after the ionization, let's move forward. Let's be quick. So we need to have another file. So let me move back to my files folder where they are. And I need to have another file, which is em.mdp. So this em.mdp file will be utilized for what? This em.mdp file will be utilized for the energy minimization. So I'm going to paste it here, this file. Let me show you this file. So if this is actually going to be the file here. Now, if you look at it here, that the algorithm which is going to be used, that is going to be the steepest gradient method. So the guys, Arslan, moderators can send you this file. If you will be interested, you can ask them, or you can prepare that file by reading the Gromacs tutorials or Gromacs documentation. But I hope so that they will provide you. So this is actually going to be the file. These are going to be the parameters which will be used during energy minimization. So what we are going to do, let's move back. Let's move back here and let's type the command. And once again, we are going to use first we are going to create a TPR file. Excuse me. We are going to create a TPR file, and to create a TPR file, we are going to type the GMX. After typing a GMX, we will type Gromap. 1 second, please. We will type Gromap. Gromap. After typing the Gromap, then we will type the -f and we will give the em.mdp. This is actually going to be the file that will be utilized to prepare the TPR file, and I've showed you earlier. Then we will type the -c. This will be just like a similar type of a command which we have used before the ionization. And we will type the name of the name of the file that is actually produced as an output of the previous step, and that is a test_solvate _ ins.gro. This is actually going to be the command, the file. Then we will type -p, and after typing -p, we will write down the name of the topology file so that we want to update it once again. After it, we will write down the name of the output file, and output file which we want to have is em.tpr. After typing this, please hit the enter button, and as you will hit the enter button, then the TPR file will be created. Once again, we got the quotation. The big data is like vagara, vagara, vagara. So etc. and etc. and etc. So we are going to have a quotation. It means that the files has been successfully created. Now, right now, we are going to perform energy minimization. To energy minimization, we are going to type once again GMX, and then we are going to use one module md_run, and then we are going to type -v. -v will what the -v will do, I will explain it to you. -v, and after -v we will type def nem, and then after we will give a space and we will type em. This is actually going to be the commands. By the way, the moderators can send you these commands if you guys request them. So after typing this one, simply hit the enter button, and you can see that ionization, energy minimization step has been started. Now keep it in mind that if you are not typing -v, then you will not have all of that output. You can't monitor the energy minimization in real time. So this will be just like this one. So you can see it here that the energy minimization step is going to be carried out. This may take some time. It will depend upon that your system capacity. It depends upon the size of your protein, and it depends upon number of other factors. So this may take some time, and during this time, the moderators,
yeah, I think at this time, it's already done actually. Yeah, but we would like to take this opportunity for a minute. Khushbat, over to you one more time, please.
Thank you, Arsalan, and I hope you guys are learning and benefiting from this session. So I'm going to share my screen once again, and I'd like to show you guys some of the commercial services that we are providing. We have a page on Fiverr through which we are providing some commercial services based on MD simulation, in which we help clients from all over the world in designing the drug, protein linking docking, protein-protein docking, molecular dynamic simulation, nanoparticle simulation, virtual screening, and QSAR analysis. We also provide our services in RNA sequence analysis, where we help them in data fetching, doing the quality check, sequence alignment, DSEC analysis, and network and enrichment analysis. Our services also include omics analysis of metabolomics data, lipidomics, proteinomics, and we also help in NGS sequence analysis. And you guys can contact us through our email that I have provided in the chat, and also through our LinkedIn page, and also through Fiverr, and all of the URLs of these networks have been provided in the chat. This is it from me, Arsalan. Dr. Naeem can continue. Thank you.
Okay, so let's be very quick so that we can answer. We could have an adequate time to answer your questions. So after this, after starting,
Dr. Naeem, you need to share your screen one more time, please.
Oh, so sorry. I think so that my screen is not shared yet. Okay, so let me share my screen. So our energy minimization is done, and you are going to have these values. So by looking at these values, we can say that our energy minimization has been successfully carried out or not. You can also plot the energy values to get an idea that the energy minimization is done or not. So primarily, the energy curve should come down. So anyhow, the energy minimization is done. Now this is time for the equilibrations. So once again, for the equilibrations, first we will perform the NBT and then NPT equilibrations. And once again, we need to have the MDP file for them. The nbt.mdp and npt.mdp file, the two files we need to have. So the two files which we need to have, let me fetch them. Let me fetch them from the previous one. So we have this NBT and NPT and files. So let me come back to my folder. So I'm going to place them here. We can see that NBT and MDP files. So let me open them into a notepad once again, so you can see the content of these files. You can prepare it by yourself, or you can ask the moderators that they may send it to you guys if you really need it. So we are going to use these files. So let's move back, and now this time let me be quick, and I'm going to edit this one, the already typed command. So here nbt.mdp, and here we are going to use em.gro. Em.gro is a file that will be created in a previous tab. And here I will type nbt.tpr. So this is actually going to be the command that will create a TPR file for NBT calibrations. So let me hit the enter button. And as I will hit the enter button, so I got an error. What I got an error? I think so there's something wrong. So that C option is, sorry. That C option is something missing here. Okay, sorry, dash R. We also have to provide one more option. That's a restrain option here, dash R, and once again .em.gro file here, and I think so we are done. Let me run it, and I hope so that this time we are successful. No, what happened? Okay. This is not an appropriate. Arslan, can you please send me the NVT file on my WhatsApp, please?
Yes, sir. 1 second.
Give me just 1 second, please. 1 second. I'm actually lacking the file. Give me just 1 second, please. I'm sorry for that one. Have you sent it?
Just a minute.
Should I bring it from? Okay, I got it. Also, please send me the NPT and MDP files as well. Okay, NPT. Okay, we got it file. I got the file. I got this file as well. So let me show Sorry, guys. There is a I've got the wrong files. There was a bit wrong in the files. Okay, so let me share my screen with you once again. Share, come on. Okay, so I'm actually going to type a similar command, but there was some issue regarding to the content of that NVT files. I used the wrong one. I basically previously I used a NVT file that is for the ligand protein simulations, not for the whole protein simulations. So right now, as you can see it here that our NVT first step has been successfully done. Our TPR file has been created. Now we will type this command gmx md run -v -defnam, and then we will type the NVT here, just like the previous one, energy minimization, and we will hit the enter button, and our energy minimization step has been started. You can see it here that we are going to perform the NVT equilibration for 100 picoseconds, and total 5,000 steps will be carried out. So obviously on my system, this is also letting me know that this is going to take approximately half an hour to complete this equilibration. And just like for NVT, this will also take a half an hour for 100 picoseconds. And for production, same, like we are going to use a similar type of the commands, nothing different, just an input files of the previous step will be output file of the previous step will be used as an input file of the current steps. And then we will move for NPT and the production steps. But obviously, this is going to take a good amount of the time. And obviously, right now, we don't have so much amount of time obviously, you guys your time is very precious. So what we have done, we have already, before this webinar, we have already fetched the results for you. We have already fetched the results for you, and you can see it here that the area, the gyrate, hydrogen bonds, RMST, and RMSF ones. So these are the graphs. You can see it here that these are the results which we have fetched from WIT. Normally, I carried out the energy minimization up to the in my system, and then after preparing that whole folder, I bring it to the supercomputing facility. Right now, or you can see here my supercomputing facility, and this system is basically RTX 4090, and I'm really thankful to the Bioinfo expert for collaborating with me. This is their system. I'm just collaborating with them, and they are providing this facility for my academic activities, so I'm very much obliged for them. So I brought all of these files here, and then after bringing all of these files, we will run the production phase, NVT, NPT equilibrations, and etc. and etc. So let me show you here the XTC file. Where is XTC file? Where is XTC file? So all of the steps has been done here. So the way will be an XTC file. Where is XTC MD? I think so somewhere here. Yes. Here you can see the XTC file. If you can see it here, that this is an XTC file, and this is just a 30 amino acid protein. And if you look at the size of this XTC file, so you will find it out that this XTC file will be approximately 800 MB file. And just keep it into mind that these are just a 30 amino acid protein. So imagine it by yourself, if you have a protein of a 400 amino acid or 500 or 1000 amino acid, then how big it will be. So just the commands which I have shown you earlier, you will use a similar type of command to carry out the NVT and then NPT, and after this one, you will use a similar type of the command to carry out the production phase. But the one very important thing for the production phase, which will please let me tell you.
Dr. Naeem, maybe you can show this one-time energy minimization and just one NPT on this supercomputing facility just one time. I hope it's not going to take a long time, but I think it will give a general better idea how these things will go.
Okay. So let me perform the energy minimization, NPT steps here. So I'm going to the open terminal. And in terminal, I'm just going to copy and pasting the similar type of the commands. I hope, Sir Arslan, you can help me out in this one, please. If you can copy and paste those commands, please.
Yes, sorry.
Arslan, can you please copy and paste those commands here?
Yes, of course.
So first copy the NVT1 and then NPT1, please. First prepare the Gromac file and then carry out the MD once. These guys are, let me tell you, the participants that these guys are doing a wonderful job. So if you need any type of the services from them, so then please don't hesitate to contact them. So please don't hesitate to contact them. I hope so that they will provide you the state-of-art services. Okay, so right now they have a very good system right now here, RTX 4090, the supercomputing, very powerful GPUs. So let's hit the enter button, please.
Yeah, Dr. Naeem, you can continue from here, I guess.
Okay, so I'm going to hit the enter button. But sorry, light is turned off at my side. Let me hit the enter button. Okay, so the NVT steps has been, NVT is carried out, and you can see it here that on my system, it was taking approximately half an hour time. And in here, in this system, this is going to take just 0 seconds. And you can, if you see here, the output of this system, that's amazing. This is amazing. So you can see it here that this system can actually carry out 70, for this protein, this can carry out the simulation of 774.776 nanoseconds per day. So if you want to perform a 1000 nanosecond simulation on this system for this protein, so this should be done within about just 1 day. So this is a very, very powerful system. So I hope so that now you will have a good idea about it. So let's come back, let's move to the one very important things here. And this very important thing is the content of the MDP file. So let me download the MDP file from the Salon that's just sent me. Okay. So in the MDP file, the one very important step is actually what? Is actually this one, the N steps. Now, using these N steps, you are going to do what? Using these N steps, you can change the time of simulation. Right now, if you see it here that we are using these number of the steps for the 50 nanoseconds. But let's say that if you want to perform a 100 nanoseconds, so then you have to make a change here. So you will make a change here, you will make this number double, and when you will make this number double, so rather than a 50 nanosecond, you will be able to perform a 100 nanosecond simulations. So this is actually going to be the most important thing. The rest of the content, if you are a beginner here, so then the rest of the content is not so much important for you. You just need to be careful at this particular one step. So I hope so that you will have a good idea about that how you can perform the molecular dynamic simulations using the Gromacs. Finally, come to the results, and in the results, you will have the idea about the RMST. So let me show you that. How you will open the RMST files. So to open the RMST files, these RMST files will be created in an XVG format. And to open the XVG format, we are going to use an Excel normally. So I'm here in my Excel. I will open, then browse, and I will navigate to the folder where my files are located. So it's a result section. I will click here on the all files so that Excel can detect them. Here is an RMSD, then delimited. After delimited, I will click next, then I will click a space, then I will click next, and then finish. And here you can see the results, the RMSD figures. So this is going to be the time, and this is going to be the RMSD values. So to plot them, what I'm going to do, let me name them. So this is RMSD. So let me move ahead. So let me check both of them, and then let me come to the insert, and in the insert, I will click here, and you can hear the RMSD values of our protein. You can see that how much this protein has been changed over the period of time. I hope so that this will be clear to you. Obviously, due to the shortage of time, I can't explain you the rest of the other things. It will be a bit difficult for me. So this is all from my side. If you guys have any questions, sorry, my camera, you can see the black here because light is turned off here. It's a bad weather here. So I hope so that everything will be clear to you now. In case of, if you guys have any question, I will be happy to answer your questions, please. Over to you, Arslan and Pushpa.
Yes.
Over to you, Arslan.
Yes. So thank you very much, Dr. Naeem. That was actually very informative for me as well. I also learned a couple of conceptual parts as well. So please, we are open for questions as well. But I think maybe it's better if we start answering a couple of the questions which we have left behind in the previous section.
Okay.
Okay. Okay, so I'm looking at this. After downloading the PDB code of my target, how do I know if there are any missing residues? How should I check this?
To check the missing amino acid residues, you need to open your PDB file into the notepad. So let me show you here. That's a good question. So let me come here to my protein, this one. So this is my protein structure. So right now you can see that you bimol sign here. But if you want to check the missing residues, amino acids, so you need to right click on it and you need to open into the notepads. So when you will do that, so the content of the PDB file will be appearing in front of you. So here just type Control F and write missing. So if there will be any missing residue, so then they will come in front of you. Right now there isn't any missing residue, so that's why I'm having an output that cannot find missing. So if there will be any missing residues, so then they will come in front of you. You could have an idea that these amino acids are missing in my protein structure. I hope so that this will be clear.
And the follow-up question after that was, and how to repair the structure, as they can be how to repair the structures if you find any missing, and of course there could be hundreds of atoms.
Okay, so if you want to repair your protein structure, so then normally we are going to use a modeler. Obviously right now there isn't a time to explain that how we can use a modeler to repair our protein structure, but on your YouTube channel there is a 1 video tutorial which basically talk about that how you can use the modeler to repair your protein structure. So if our participant can watch that video tutorial, or they can join in our courses, so I hope so that he will learn in a better way how to prepare or repair the protein structure. The YouTube video is there for this one.
And there's also a question next is it necessary to make solvation for a complex protein or DNA organic ligand to get a stable structure, and what is the appropriate force field that I have to use in that case?
Please remember that there are the two different type of molecular dynamic simulations. The one is an implicit, another one is explicit. Sometimes we need to put our molecule in the water box, but sometimes this is not mandatory that you are going to put your molecule into the water box. So if your study question is going to be answered without putting the water molecule, without putting your molecule into the water box, so then it's okay, you can move ahead. So solvation is not a mandatory step. That depends upon your study question. What is your study question? What type of molecule you have? So normally in case of protein, we can't move without solvation. But if you have an organic molecule and you want to just check its properties, so it's totally fine. You can move ahead with it. You can go ahead without solvation. Can you please make everyone mute, please? There is noise is coming.
Let me see. Probably Mohammed Mudassar, I guess. Yeah. Now I think it should be fine.
Okay. Thank you. Okay, another question, next question.
Yeah, so the next question is from Sonia, and she's asking, we are doing simulation for some molecules and getting some results. How do we know that the results we get are right?
It's a very generalized question. I must say that it will be very hard to comment about it, that how you can say that your results are right. It totally depends upon your study question, your study design. Let's say that during my PhD, I was interested to see that due to the mutation of one amino acid at one particular position at my protein, I'm interested to see that is this going to be, protein is going to be stabilized or that protein is not going to be stabilized. So I did a molecular dynamic simulation of my wild type protein and the mutated protein, and I compared the results. And then I got an insight that my protein is getting stable or my good protein is getting unstable. So how to interpret the results? That depends upon your hypothesis. Obviously, Sonia, I need to look at your results before making any final comment that your results are right or wrong. And I also need to know your study question before commenting on this question. And
Yeah,
I'm sorry,
exactly. It depends on
that's
the
what
kind
I
of the
can say.
mutation, where you exactly did the mutation. So if it's in the
Yes.
hotspot region, so you would probably have a
Yeah.
hypothesis what exactly are you going to be expecting, and
Yeah.
if it's
If my
divided,
protein,
yeah,
if my protein is unstable, so I'm expecting the higher RMST
yeah,
values. I'm expecting the high radius of gyration, the high solvent accessible surface area, low number of the hydrogen bonds over the period of time. So this is giving me a hint that my protein is getting unstable because of the mutation.
yeah.
So it depends on the study question that what you have and what type of analysis you are going to carry out. For example, in case of the ligand protein interactions, sometimes RMST is not going to give you a good insight. You need to calculate the delta G values. So for every type of study, for every type of question, you need to carry out some different type of analysis. And obviously you need to go through the literature to answer those questions.
Sometimes, of course, the interaction energies have to be calculated, or that we have to
Yes.
go for them in
Yeah.
PBSAs. There could
Yes.
be, of course, many steps that we can take
Yes.
to
Yes.
ensure if the system is working properly.
Yes.
Yeah. And the next question is, how can we know that the energy of the given structure has been minimized successfully?
Yeah, that's a very nice question. I showed some of the values at the end of the energy minimization. So these values basically help you out to get an idea about that your system has is successfully energy minimized or not. Another thing is that you can actually make a plot of your energy values. So Gromacs have an option for you where through which you can actually plot your energy values. And as I told you earlier during my practical demonstration there, the energy curve should come down should show the downward trend. So if your energy curve is showing the downward trend and up to the zero, towards the zero, then this is actually a good sign that your system is properly energy minimized. But you need to plot it, and the Gromacs have an option to plot it.
Exactly.
You can easily plot it there.
Right after
But if
you
you
do
will
the
check
energy
the
minimization, there is a
yes,
step of making a graph, and you can have a look.
Yes,
Yeah.
you can make, there is a step of making a graph. You can check the Gromacs documentation to find it out at this command. And obviously that was not we don't have enough amount of the time, so that's why I just skipped it.
Yeah. Okay, so the next question is how we will know different drugs' relative interaction strength with a protein?
That's a very nice question. So right now our focus was just on simple protein simulations.
Yes.
We haven't studied we haven't have the protein right now. Maybe in a next webinar, we can actually let our participants know that how they can actually do the simulation of a drug and the protein.
Yeah.
So if you're actually simulating your drug and the protein, so in this case, you need to calculate the three very important things. Number one, you need to calculate the RMST value of your drug with the protein. The second very important thing is that you need to calculate the interaction energy of your drug with the protein. And the third very important thing is that you need to calculate the delta G value of your drug with the protein using MMGB-assay module. And another very important feature, another very important thing, I must say that here, I must quote it here, that is the total number of the hydrogen bond that your ligand is going to make over the period of time with the protein. So these are the normally 4 different type of the parameters in which we are really interested to get an insight that the ligand is strongly bounded with the protein or the ligand is weakly bounded with the protein. So through these 4 parameters, we could have an insight about the binding intensity of the ligand with the protein.
Okay, so at this point, I would, before moving forward, I would like to ask Afiz, because he has been raising his hand for a long time. I would like to give him the time to ask the question if he has any.
Yeah, sure, please.
Okay, thank you so much, Professor. This is a very great presentation. My question goes thus. I am not a biology person, so I am a physical chemist, and then what I am actually interested in is I am studying the non-covalent interactions between some molecular systems. And my interest is to use some spectroscopic study to investigate these non-covalent interactions. And I read some paper. So when I read this paper, I saw that people have actually used Gromacs, or let me say molecular dynamic simulations, to predict the, let's say, for instance, Raman spectroscopy of some system. So, and from what I get from your presentation, you guys are working on, let's say, for example, you are working on protein, and then before you start the simulation, you have to get the protein, maybe from a protein data bank, right?
Yeah.
Okay, now, so let's say I have a new system which does not come from maybe protein or maybe some biological materials. So how can I design this new system for me to supply, to get an input file that I will use for the molecular simulation dynamics?
Okay. Thank you so much, Hafiz, for your nice question. And first of all, I'm very much thankful for your kind words of appreciation. If this effort is useful for you, so I think so that we have done a lot. So we just want to contribute. We just want to extend the knowledge, and we want to contribute for the betterment of the society. So if you learn something, so this is a huge achievement for us. So thank you so much for your kind words and kind appreciation. Now let's come to your question. Let's say, assume for a moment that you have some molecule. So you can actually draw the three-dimensional structure of your molecule by using a number of the different softwares which are available, which are specifically designed, keeping the chemistry persons in need. The one software is a ChemDraw. You can use a ChemDraw, and you can draw the three-dimensional structure of your protein, of your molecule. Similarly, there is another paid software that is a Material Studio by Bovia. You can use that one, and you can also draw the three-dimensional structure of your material there. But these two are the paid softwares, and obviously I'm not advertising these paid softwares. I'm just letting you know the options. Along with this, there is also one software which is known as ChemSketch. The ChemSketch is freely available, and you can use that one, and you can actually draw the three-dimensional structure of your protein in the protein of, I'm saying protein because I'm a biology person, so I'm repetitively saying the protein. So you can draw the three-dimensional structure of your molecule there in the ChemSketch, but ChemSketch is freely available. I think Hafiz is gone. Hafiz is there. Okay.
Yeah.
Okay. So once you have a three-dimensional structure of protein and you know the nature of your molecule, so then by looking at the nature of molecule, you can decide that which type of the force field will be most suitable for you. Let's say that it could be an charm force field may be suitable for you, but normally the charm force field are designed keeping the proteins in view. For the for the pure chemicals like molecules in your case, you need to first find it out that which type of the force field are suitable for you. As you know that I'm not a chemist, I'm a biochemist, and I'm actually more interested towards the protein side. So that's why my literature review is primarily confined in a protein area. So I don't exactly know right now which type of the force field will be suitable for you, but you can search it out.
Exactly. This is actually, sorry, I would like to convene here as well. That most of the molecules are also findable on ChemSpider as well, or is it SpiderChem?
Yes.
I don't know. ChemSpider.
Yes, it's a ChemSpider.
ChemSpider, yeah.
The three-dimensional ChemSpider, if that is, but He's is saying that his molecule is completely novel.
Okay.
So his molecule is completely novel. He can actually draw a structure in a ChemSketch. So you can decide that which force field is most suitable for you, and then you can use that force field. You can bring a folder of that force field into the working directory. You can put the three-dimensional structure of your molecule in the working directory, and then you can start moving ahead. If solvation is needed, you can do the solvation. If solvation is not needed, just minimize your system, carry out the energy minimization of system, equilibration, and then production of the system. And once you have the XTC file, after the MD simulation, then you can perform the specific type of analysis in which you are really very much interested. I hope so that it will help you out.
Yeah, that's Thank you so much for I have one follow-up question again, please. Okay. Now, from the results you presented, so from my question, I said what I'm actually interested in is to get spectroscopy properties of the system, right? So let's say is it that I will tell Gromacs to give the output of what I'm actually interested in. For example, let's say I want to see the Raman
Raman effect. Okay, Raman spectroscopy. So actually, Hafiz, there are many different modules that don't come with the Gromacs by default. You need to actually install them into the Gromacs. Once you install the Gromacs, then you need to install those particular modules in which you are really very much interested. For example, in case of the Raman effect, I'm pretty sure that to calculate the Raman properties, the Raman effect, there will be specific modules in a Gromacs that will be available. Somebody will have developed it. So you need to install it and then you need to plug it with your Gromacs. And once you will plug it into the Gromacs, then you can use the XTC file in those modules to calculate your results in which you are particularly interested.
Okay, thank you so much. Yeah,
and of course, just to plug in, we can actually help you with these kind of things as well, because this is what we do in Bioinfo Expert. So moving on, we have, I think, Dr. Naeem, if you would like to please keep it a little bit crisp. I'm so sorry because we're already beyond our gazetted time for the participants, and of course I can imagine it could be a bit time taking. So I will just take a couple of more questions, and probably I'm so sorry for the rest of the questions. Maybe you can just email us, and we would be very, very happy to help the rest of the participants, of course. So maybe just two more questions. I'm just going through. Okay, for the MD simulations on materials like MOFs, can you please specify force field? I'm struggling to apply force field in the ADF. I applied CU BTC, but still didn't work.
I'm sorry, there was a bit lag and a bit distortion. I couldn't hear the question properly. Can you please repeat it?
Yes. So the question is, for MD simulations on materials like MOFs, can you please specify force field? I'm struggling to apply force field in the ADF. I applied CU BTC, but still didn't work.
Actually, I think so that the participant is really interested in to study the molecular dynamic simulation of certain materials.
Yeah.
Maybe it could be the nanomaterial. I don't have a good idea about these nanomaterials because my area of expertise, as you know, is the proteins. So I'm actually more confined towards the proteins. So that's why my literature view specifically for the nanomaterials or the chemical materials is very limited. So it will be very hard for me to right now comment on it. I need to look at the literature view and then we can get back to the participant.
Yeah.
You can ask them, you can ask the participant to send an email. So maybe when we will get some time, so we will respond them back.
Of course, exactly. Yeah, this is of course a very understandable problem for finding the force field.
Yeah.
So how to fix the topology error?
How to fix the topology error? Okay. Please make someone noise. It's noisy here. Okay. So if you're getting the topology, if you're getting an error in PDB to GMX module and topology errors, I'm assuming that the participant is want to ask a question related to the PDB to GMX modules, just
exactly,
like we face an error a few during our practical demonstration. So if you're not going to find out the topology of one or two amino acid residues, this is totally fine. You can just simply use the -IGNH, and the -IGNH stands for the ignore. You're asking the Gromacs that please, if you're not finding out the topology, right topology of the one or two amino acids, then you can skip it, and this is quite safe. It wouldn't harm, and it wouldn't harm too much.
But exactly, the question was to how to fix this issue. So
yeah, I'm not talking about it.
Yeah, sorry.
I'm going to answer it. So let's say that you're finding an issue of multiple different residues. I mean, 4, 5, 6, 7, 8, and 9, 10. It means that your structure is not proper. Your structure is not correct. Either your structure, you are not selecting the appropriate force field where these parameters are not available, or your structure is not correct. So you need to work on those things. You need to work on you need to work on your structure, or you need to change the force field. For example, and very recently we did a work on a lithium ion. So when we were trying to do this molecular dynamics simulation of lithium ion, so we faced problem that the charm force field was not working on the lithium ions. That was not creating a topology file of lithium ions, because charm force field has a limitation that it cannot create a topology file of the lithium ions. So what we did, we moved to another force field, and that force field was OPLS. The OPLS have the force field of all atoms. Either it's a lithium, it's a sodium, it's a potassium, it's a calcium, or whatever. So if you have facing a problem, so you can fix it in a 2 steps. Either check the structure and make it correct, or change the force field. It's up to you that what you're going to do.
Yeah. Okay. Sorry. Yeah, so I think this is going to be the same as a question as Afeez probably. I'm not sure, but you're much better to answer this. Can we use the MD simulation for the nano cages of transition metals?
Yes. The answer is yes.
Okay,
yes,
okay.
I heard it.
You read that? Okay.
Okay, I read it.
I thought maybe you become very enthusiastic about that.
Yeah, yes, you can perform the molecular dynamics simulation theoretically on all type of atoms if force field is available.
Yes.
If force field is available. And there is a very good research is going on the nanomaterials, where the nano cages and the nano composites are subjected to the molecular dynamics simulations to check their properties, the chemical properties and the structural properties as well. So yes, the answer is yes. Nano cages, you can apply the molecular dynamics simulations on the nano cages, nano composites, nanoparticles. But obviously, the first and most important thing is that you need to have the 3-dimensional structure of your nano cage and the nano composites, and be very clear that you need to prepare a very accurate depiction of your nano cage and the nanoparticles.
Yeah, okay, so I would like to also read maybe second last question. Let's say I keep saying last questions, but okay, just because the questions are very interesting, of course, for me as well. So the question is, thank you very much for providing the information about the molecular dynamics simulation, but the problem is how to perform the molecular dynamics simulation on my laptop around 100 nanoseconds in minimal time. It takes very much time, almost 1 to 2 months.
Yeah, as I told you earlier.
And of course, we are here to help you with this as well as a bioinformatics expert. We have the facilities to do these kind of things probably in hours, not even in days, which probably is going to take you months or maybe more. We can probably do the same kind of analysis for 100 nanoseconds or more in days. Without even, of course, because we have acquired the systems to specifically comply with these kind of huge simulations. So maybe just please drop us an email, and hopefully we can help you with this as well. Okay, so now just last question. I'm just going through. I'm sorry if I'm missing any questions.
I just want to add one thing. I just want to add one thing. So let me appreciate you guys that as the participants have seen that it took approximately half an hour to complete the just NBT step in my system, which is obviously not a bad one. It's a good one. But let me tell you, the participant, that you guys are providing me a very good facility where I can actually run my molecular dynamics simulations for my academic purposes. So I'm really thankful to you guys there. So if participants needs anything, any help, so definitely they can contact you. And I know that you guys are providing a wonderful services.
Yeah. Okay. So last question. Thank you.
Before Arsalan, you take the last question. I'd like to tell the participants that guys can reach us through email, LinkedIn, Fiverr, and as well as on Twitter. And the links are provided in the chat. And those questions who are not answered, you can also ask your questions through Twitter and email as well. Thank you.
Yeah. And of course, when we will be sending you these certificates, we will also be attaching a sort of a one-pager as well, so just give you an idea of the services and how exactly can we help you. So last question, and please, Dr. Naeem, if you would like to keep it crisp, that would be awesome. So if we change the force field, what's the difference between the OPLS 4 and OPLS 2005?
OPLS 4?
4, yeah. And OPLS 2005.
Actually, I don't have a very good idea. I haven't
because the force fields are very complicated to.
I haven't studied. I haven't studied the difference between the OPLS 5 and the OPLS 4, so it will be hard for me to comment right now. I need to go through the literature, and then I can answer this question in a better way.
because these force fields can get very messy. Even if you open the folder for this CHOM 36, which you have Dr. Name in front of you right now, so maybe the participants can see in the folder.
Just 1 second.
Yeah.
So this
So
is
see,
CHOM
of course,
36
things
force
can get
field,
very
and So
interesting.
if you can see it here that there are a lot of the files having a different type of the parameters. So it's need to, I am actually haven't studied I haven't used the OPLS very frequently. I normally use the CHOM force fields because CHOM force fields are fulfilling my requirements. So I normally stick to the CHOM force field and normally follow the guys who develop the CHOM force field. So I haven't studied the force field in detail.
Okay,
OPLS.
so I would like to thank everybody who was here and who has already left. I can understand, of course, but I would like to again formally thank Dr. Naeem Mahmood Ashraf for the time that he has provided us, and with the brilliant, of course, as always, with the lecture. And hopefully, if everybody would be interested, we can of course continue with a more advanced session later on. And of course, we are also planning on doing the RNA sequence analysis and the NGS space and the transcriptomic, lipidomics, proteomics as well later on in the future. So if everybody would be interested, we will definitely drop you email. And also for the molecular dynamic simulation advanced webinars as well, hopefully soon.
One thing, I think so that you guys should continue this one bi-monthly or at least once in a month.
We are actually, we will plan like this, yeah, definitely.
And I will also request all of the participants to give us a feedback regarding the quality of this webinar so that we can improve it in the future.
And again, thank you very much, everybody. And I would wish you the best of luck and the rest of a great evening. Yeah. Thank you very much, everybody.
Thank you so much.
Have a good day. Bye.
Bye-bye.
Bye.